Welcome back to the deep dive. Today we're talking about a really sensitive creature, one that tells us a lot about our freshwater environments in Europe.
That's right, the white-clawed crayfish, Ostropotamobius palipes. It's a crucial bioindicator species.
But it's in serious trouble. We're talking major declines.
Huge declines. Some estimates are between 50 and even 80% loss since the 2000s.
Hmm.
Habitat degradation, water quality issues, and this devastating crayfish plague spread by invasive American species. It's a perfect storm, really.
And checking on the populations that are left, well, the old ways just aren't great.
No, definitely not. Things like electric fishing or trapping, they're disruptive, incredibly time-consuming, and honestly not very effective. When you've got these scattered low-density populations, you can miss them easily.
Which brings us to the focus of today's deep dive, the molecular approach. Environmental DNA or eDNA.
Exactly. Instead of catching the animal, you just take a water sample. And the idea is to detect the tiny bits of genetic material they shed. Mucus, feces, maybe skin cells. It's much less invasive.
A game changer for sensitive species. So we're diving into a paper led by Thomas Baudry titled Influence of Distance from Source Population and Seasonality in eDNA Detection of White-Clawed Crayfish through qPCR and ddPCR assays.
Their mission was basically to figure out the best practices for using eDNA to find this specific crayfish in France. They looked at, well, when to sample, how far downstream you can detect it, and compared a couple of different lab methods.
Essentially creating a how-to guide for conservationists using this tech for this species.
Okay, so first, why this crayfish? What makes A. polypes such a good, well, strict indicator?
It's incredibly fussy about where it lives. It needs really pristine water, cold, lots of oxygen. You typically find them in those cleaner headwater streams.
Right, so if the water quality drops or the habitat changes, they're often one of the first species to disappear. They're like that canary in the coal mine.
Makes sense. So finding out precisely where they still are or confirming where they might have vanished from is critical for conservation.
Absolutely. In this study, they looked at 10 different streams over in Indre-et-Loire in France. They wanted to nail down how seasonality, environmental factors like temperature and oxygen, and crucially, distance from a known population affect whether you can actually detect its eDNA.
Before they even got to the field stuff, though, they had to make sure their lab tools were spot on right, choosing the right genetic target.
Foundational work, yeah. They picked a specific bit of the mitochondrial COI gene, about 96 base pairs long, and they did rigorous testing, both on the computer, in silico, and in the lab, in vitro.
Why? To make sure they weren't accidentally picking up DNA from other things.
Exactly. Especially those invasive American crayfish species that often live in the same areas. They had to be absolutely certain their tests would only light up for the white-clawed crayfish. Specificity is everything here. If you get false positives, your conservation efforts could be completely misdirected.
Okay, validation done. Then they looked at quality control, like how sensitive the tests were, the limits of detection and quantification.
Yep. They established those for both PCR methods. They compared qPCR and ddPCR. Found they were pretty similar, actually, around 4 times 10 to the -5 nanograms per microliter. Very sensitive.
Now, here's something that often trips up eDNA studies. Inhibition. You get stuff in the water, humic acids, mud that can mess up the PCR reaction and stop it from working, leading to false negatives. How did they handle that?
Well, this was one of the really interesting sort of aha moments of the paper. They tested for inhibition and basically found none.
None. In environmental water samples. That seems unusual.
It does, but it makes sense when you think about where these crayfish live, those pristine, cold, clear headwater streams. They just don't have high levels of those typical inhibitors. The maximum inhibition level they measured was tiny.
So the crayfish's preferred habitat actually helped make the eDNA method work better.
Pretty much. It meant they could be really confident in their detection results. They weren't getting false negatives because of gunk in the water interfering with the test. It was a huge advantage for this particular study system.
Okay, so with that clean signal, they could then look at what environmental factors actually predicted finding the crayfish eDNA. What did they find?
The results lined up perfectly with the crayfish's ecology. They used occupancy modeling, a statistical approach, for both PCR methods. Higher oxygen was a big plus. Detection probability jumped to 75% when oxygen was above 8 milligel.
And temperature.
Temperature had a negative effect, as you'd expect for a cold water species. Detection was best, over 80% probability, when the water stayed below 20 degrees Celsius. It just reinforces that eDNA is reflecting the actual habitat needs.
Right. Cold, clean, oxygen-rich water. Now, what about that finding on the volume of water filtered? That seemed a bit counterintuitive. They got better detection when they filtered less water, under 1,000 milliliter.
Yeah, that sounds backwards, doesn't it?
Oh,
You'd think filtering more water would give you more DNA.
right. More water, more chance of catching those shed cells.
But think about why you might filter less water before the filter clogs. It's usually because the water is clearer, less turbid, with less suspended sediment or organic matter.
Ah, so filtering a smaller volume wasn't the goal. It was an indicator that the water was very clear.
Exactly. It indicated they were sampling in those pristine headwater conditions that the crayfish love. If your filter clogs quickly after filtering a large volume, it probably means the water is murkier, maybe downstream, maybe less suitable habitat. So if filtering less wasn't causing better detection, it was correlating with the high-quality water where the crayfish were likely present and shedding DNA.
That's a neat ecological link. Okay, let's talk distance. DNA washes downstream, but it degrades. How far away from a known crayfish population could they reliably pick up the signal?
This is super practical for monitoring. Their models showed a pretty significant drop-off with distance. Using standard qPCR, the detection probability fell below 60% once they were sampling more than 1 kilometer downstream. For ddPCR, it was below 50% beyond that 1 kilometer mark.
So about a kilometer seems like a reasonable radius for high confidence.
Generally, yes. But, and this was encouraging, they actually managed to get detections much further out in some cases.
Really? Because I thought crayfish, with their hard exoskeletons, maybe don't shed as much DNA as, say, slimy fish.
That's true. They are thought to be lower shedders, which makes the fact they could detect eDNA up to 2 kilometers downstream in a couple of streams, Lelong and Rorth, quite impressive. It suggests eDNA could still be useful even for these less sheddy, low-density populations.
2 kilometers is quite a reach, but it wasn't consistent.
No, definitely not. Stream dynamics play a huge role. In another stream, Quintain, the detection probability crashed to just 13%, only 300 meters downstream. So things like flow rate, water volume, maybe sunlight exposure causing DNA breakdown, they all influence how far the signal travels. You can't assume 2 kilometers everywhere. Okay, moving on to another key factor. Seasonality. Does when you sample matter?
Big time, apparently. They tested in June, August, and October back in 2021, and they found season had a significant positive effect on detection.
And the winner was?
August. Detection probability really peaked then, hitting over 90% with the qPCR method.
And there's a good reason for that August peak, isn't there? It's a combination of factors.
Yeah, two main things seem to be at play. First, the crayfish themselves are likely more active in the summer, maybe more movement, more feeding, and importantly, molting shedding that old exoskeleton releases a bunch of DNA.
Right, higher biological activity. And the second factor?
Hydrology. August is typically when water levels and flow rates are lowest in these streams, naturally.
So less water means the eDNA that is being shed gets less diluted, it's more concentrated.
Exactly. High shedding meets low flow. Perfect conditions for detection. It really underscores that you need to think about both the animal's biology and the river conditions when planning sampling.
Okay, now for the lab method comparison. The classic workhorse, qPCR, versus the fancier, potentially more sensitive digital droplet PCR, ddPCR. What was the verdict?
This was another surprise, I think. Given that ddPCR is often touted for its ability to detect really low amounts of DNA and resist inhibitors, you might expect it to outperform qPCR here.
Especially in environmental samples.
But they found no significant difference. Statistically, the detection efficiency and probability were basically the same for both methods.
Wow. So ddPCR didn't offer an advantage in this case.
Nope. And we know why, don't we? It goes back to that earlier point about inhibition.
Because the samples were so clean.
Yeah.
ddPCR's major strength is often its better performance with inhibitors present, or when quantifying really tiny amounts. But if you don't have inhibitors,
then that advantage disappears. Both methods could perform at their best, and in this system, they turned out to be equally good at just saying yes or no to crayfish presence.
that has some pretty big practical implications for managers then. If performance is equal, what's the recommendation?
It comes down to cost and time. qPCR won handily on those fronts.
And probably cheaper.
They estimated qPCR analysis was around 17.60 euros per sample compared to 30.50 euros for ddPCR.
Yeah.
That's a substantial saving when you're potentially processing hundreds of samples.
And
qPCR is faster. You skip the whole step of generating the tiny droplets needed for ddPCR, which takes maybe 30 minutes or more per batch. Plus, qPCR results often come up in real time, while ddPCR needs a final read at the end. They reckon qPCR saved over an hour in total per run.
So the clear takeaway for conservation groups working in similar clean water systems is stick with qPCR. It's cheaper, faster, and does the job just as well for presence-absence detection here.
So pulling it all together, we have this optimized eDNA approach. Sample ideally in August, focus within that 1 to 2 kilometer downstream zone, and use the cost-effective qPCR method. How does this actually help white-clawed crayfish conservation on the ground?
It's a powerful tool for strategic monitoring. Managers can use eDNA as a first pass, a rapid screening tool across many sites relatively cheaply.
To sort of map out potential hotspots where crayfish might still be hanging on.
Exactly. You get a much better idea of their potential distribution. Then you can deploy the really resource-intensive traditional methods like physically going in to count them, assess population structure, or collect samples for genetic analysis much more strategically. You focus those efforts only in the areas where eDNA suggests they are likely present within that 2-kilometer detection window.
So it doesn't replace traditional surveys entirely, but makes them vastly more efficient.
Precisely. It guides where you invest that heavy fieldwork effort. And sadly, the study also showed its power in confirming absence.
Yes, the Brule 2 station example.
Yeah, unfortunately, their eDNA surveys between 2021 and 2022 confirmed that a historically known population there had disappeared. It's grim news, but getting that rapid confirmation is vital.
Why vital? Because it stops you wasting resources looking for something that's gone.
Partly, yes. But it also triggers urgent questions. Why did they disappear from that specific spot? Was it a pollution event, habitat change, disease? Understanding the cause of local extinctions is crucial for protecting the remaining populations nearby. eDNA gives you that early warning, or in this case, confirmation.
Looking ahead, the authors raise a really important point about climate change. This optimal sampling window, they identified low flow, temperatures below 20 degrees C that might become less predictable, right?
Absolutely. As we see more extreme weather, more intense droughts, or shifts in seasonal temperature patterns, that ideal August window might shrink or shift earlier or later in the year. Sticking rigidly to a calendar date for sampling might become unreliable.
So managers need to be more adaptable.
They have to be. The recommendation is to move away from calendar-based sampling and towards conditions-based sampling. Monitor the stream conditions, particularly temperature and flow, and aim to sample when you hit that sweet spot whenever it occurs that year. Keeping below that 18-20 degrees C threshold seems key.
It adds another layer of complexity to long-term monitoring programs.
It does. It means you need good environmental data alongside your eDNA data. But it's essential to avoid getting false negatives just because you sampled at the wrong time relative to the conditions that year.
And finally, the potential to use this approach for other species.
Oh, definitely. The whole process they went through, validating the primers, checking inhibition, comparing methods, assessing environmental factors, it's a solid template. It could be adapted fairly easily to develop robust eDNA monitoring for other crayfish species or even other aquatic organisms of concern. It streamlines the path for applying eDNA effectively elsewhere.
Okay, so to quickly recap our deep dive on the Baudry paper, eDNA is a powerful tool for monitoring the endangered white-clawed crayfish. We learned detection works best within about 1 to 2 kilometers downstream, peaking in August due to high activity and low water dilution.
And importantly, in the clean headwaters these crayfish prefer, the cheaper, faster qPCR method works just as well as a more complex ddPCR for presence-absence, making it the practical choice for managers.
It allows for targeted, efficient conservation efforts, but it also highlights the need for flexible, conditions-based sampling in the face of climate change.
Which leads us to our final thought for you to consider. We've seen how tightly linked eDNA success is to these specific environmental windows: low flow, low temperature. As climate change makes these windows increasingly unpredictable, shrinking or shifting each year, how can conservation managers realistically maintain consistent long-term monitoring programs? How do you compare data across years if the optimal sampling time bounces around? It seems effective monitoring in the future will rely as much on accurate environmental forecasting as it does on the molecular biology itself.